welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper. This is the morning shift or the shift 1 question paper, and I am discussing the part C section. And in this section I will be discussing applied biology questions as well as methods in biology questions. So this was conducted in March 2025. So coming to the first question. So in an experiment, FITC-CD4 and PECD8 were used to stain thymocytes. So these are some fluorescent probes that are used to stain thymocytes. So they are staining CD8 and CD4 thymocytes. The cells were run through a flow cytometer and the data were plotted as CD4 versus CD8. So you have a flow cytometer and the data being plotted as CD4 and CD8. The results are shown in the figure below and the following statements are made. So which of the options represent the combination of all correct statements? So as per this question, you can see that it is a combination of the flow cytometer and the T cell maturation stages. Okay, that is a combination between your immunology and flow cytometer. So before coming to the question, we will see the T cell maturation. Okay. So you know the T cell maturation, it has got stages like there is the double negative stage, then you have got the double positive stage, then there is positive selection, and then there is negative selection, and finally the single positive stage or the maturation stage. So what is happening at each of these stages? So the T cell precursor, they migrate from the bone marrow to the thymus for maturation. So in the thymus you have got the double negative T cells. They are called double negative because they lack both the CD4 and CD8 markers. So you represent them as CD4 minus and CD8 minus. And in this stage, your pre-TCR, that is your T cell receptor, the B chain rearrangement take place. Okay, in the double negative state. Then you have got the double positive stage where the T cell double negative lymphocytes, they starts to express both CD4 and CD8 markers. And in this stage there is the rearrangement and expression of the TCR alpha chain. So in this stage you have got in the double negative stage there is beta chain rearrangement, and in the double positive stage you have got the alpha chain rearrangement. Remember these two. Then there is the positive selection. So positive selection is the T cells that recognize self-MHC molecules. And their recognition of the self-MHC molecule is essential for antigen presentation. So you select the T cells that can weakly bind to the self-MHC molecule, and the T cells which fail to bind, or if they bind strongly to the self-MHC molecule, they will be eliminated. Okay, so in positive selection, the T cells must bind to the self-MHC molecule, but not strongly but weakly. Only those which bind weakly are selected. This is for antigen presentation. Then you have got the negative selection where this positive selection and negative selection take place in the medulla. So negative selection where T cells are presented with the self antigens. So if the T cell binds strongly to the self antigen, they are avoided. So autoimmunity can be prevented. And T cells, if they bind weakly to self antigens, they are selected to become mature T cells. Then there is the single positive stage and maturation. So after this positive and negative selection, these thymocytes, they differentiate into single positive cells. That is, till positive negative selection, they are double positive. That is, they have both CD4 and CD8 markers. So some of these T cells, they after undergoing positive and negative selection, they lose their CD4 plus marker and they develop into CD8 plus cells. And some T cells, they lose their CD8 plus marker and they develop into CD4 cells. The CD4 helper cells again differentiate into helper Th1 and Th2. So this is the general rearrangement that is taking, T cell maturation that is taking place in the thymus. Then there is another T cells, that is, they express the gamma delta T cell receptor instead of the alpha beta T cell receptor. So alpha beta T cell receptor is found in majority of the T cells, but there are some T cells that express the gamma delta T cell receptor. So this gamma delta T cell receptor gets differentiated during the double negative stage, that is over here, that is in the thymus, that is in this stage, that is the double negative stage. They get differentiated into gamma delta T cell, and this expression of the gamma delta T cell receptor chains precedes the expression of the T cell receptor alpha beta chains. So first, during the double negative state, the T cells which are destined to become, destined to have the gamma delta T cell receptor, they will differentiate out from the double negative T cell progenitor even before the T cell progenitor start expressing the alpha beta T cell receptor. And these gamma delta T cell receptor T cells, they exit the thymus and they develop and mature elsewhere. So now come to the question. So before coming to the question, I will tell you how to read the flow cytometer data. Okay, now you have got the x-axis with the CD8 and the y-axis with CD4. Now it is divided into 4 quadrants. So if you see this quadrant, that is the lower right quadrant, it contains, this lower right quadrant, it will always contain events that are positive for the x-axis marker. Okay, here it will be events that are positive for the x-axis marker but negative for the y-axis marker. And this quadrant, it will have events that are positive for both x and y axis, and this quadrant, it will have events that are negative for both x and y axis, and this quadrant, it will have events that are positive for the y axis. So accordingly, I have mentioned here, that is, I told you here the events are positive for x-axis. So CD8 plus cells will be fluorescing here and CD4 minus cells will not. Whereas here both CD4 minus and CD8 minus cells will not fluoresce. Here they both will fluoresce and here only CD4 plus cells will fluoresce. Okay. So that is how you read the flow cytometer data. Now start reading the options. Rearrangement of TCR beta locus is initiated in cells included in the quadrant containing 3.58% of the population. That is 3.58 is shown in this quadrant, and I showed you this quadrant is negative for CD4 minus and CD8 minus. So they are having CD4 minus CD8 minus cells. So those cells will show beta chain rearrangement of the TCR. I already told you beta chain and rearrangement take place during the double negative stage. Okay, so this is correct. So A is correct. Then next one, TCR alpha locus rearrangement occurs in cells included in the quadrant containing 87.5% of the population. So that is here. So T cell alpha rearrangement, I already told you, it is taking place during the double positive stage. So double positive stage is CD8 plus CD4 plus. That is double positive stage, and here it is 87.5%. So that correlates with the statement given in B. So B is also correct. Then FIT CD4 and PE CD8 cannot stain the same cell. How is it possible? Then how did it stain here in this quadrant? CD8 plus CD4 plus. How did it stain there? So that is not correct. Then rearranged TCR gamma delta receptor is expressed on 7.06% of population of cells. That is this quadrant. That is this quadrant. So that quadrant is having CD4 plus cells and CD8 minus cell. So T cell gamma delta receptor is rearranged during the double negative stage. So CD4 minus CD8 minus. So it should have been here. That rearrangement of the gamma delta receptor is taking place, not here. So that statement is incorrect. So A and B only, that means 1 is the answer. The others are all wrong. A, D, A, B, C, B only. So that is also not correct. Then next one. Following statements are made with respect to the production of transgenic plants. Auxin can be used as a negative selection marker. Okay, now what is this positive selection marker and negative selection marker in transgenics? So positive selection markers, you all know, they are used to select transformed cells from untransformed cells. So you have got an antibiotic resistance gene like NPT2. And NPT2, you know, it confers kanamycin resistance. So transformed cells will have the NPT2 gene in them, which they got from plasmid, and the untransformed cells, they will not have the NPT2 gene in them. So the transformed cells will grow in a medium containing kanamycin because NPT2 will detoxify the kanamycin for them. NPT2 is neomycin phosphotransferase 2. And the non-transformed cells, since they don't have NPT2 gene in them, kanamycin will be toxic for their growth and they will die out. So here that is positive selection. Now here the question is for negative selection marker. So negative selection marker, what they do is they will either inhibit the growth or kill the transformed cells which carry these negative markers. So only cells that lack the negative selection marker in them will be able to survive and proliferate. Okay. So that is the basis for negative selection marker. So first statement, that is auxin can be used as a negative selection marker in plants transgenics as it can be lethal to germinating seedlings at higher concentration. Yes, auxin, you all know, it is a plant hormone and if at higher concentration it can become toxic, so it can be used as a negative selection marker. Okay, so A is correct. So that means 1 we have to see and 3 also we have to see. So C and 3 and 4 option gone. So A is correct. Then agrobacterium inserts tDNA at random locations, of course, in plant genome, and thus it cannot be targeted to a desired location. 100% correct. Because of its insertion in a random location in the plant genome, it cannot be targeted to one desired location. That means if you want the plant transformed for one particular character and you want the character expressed in the leaf, means you cannot use agrobacterium transformation methodology, because it will be found everywhere in the whole plant. It will be found in the stem, it will be found in the leaf, it will be found in the flower, root, everywhere it will be found. So you cannot use agrobacterium for transforming plants if you want to target to a desired location. So B is correct. Okay, now C. Okay, so that makes, I think, 1 is wrong. Okay, the antibiotic kanamycin interferes with the cytoplasmic ribosomal protein synthesis machinery. No, it doesn't interfere with the cytoplasmic ribosomal protein synthesis machinery, but it interferes with the chloroplast and mitochondria ribosomal protein synthesis machinery, not cytoplasmic ribosomal protein synthesis machinery. So that makes option C wrong and 1 also wrong. So option 1 is wrong. So 3 is the answer. Then gene transfer by ballistic or particle gun bombardment usually result in lower transgene copy number. No, it results in higher transgene copy number and less DNA rearrangement. No, greater frequency of DNA rearrangement than agrobacterium mediated transformation. So that means D also is not correct. Oh, the question is for incorrect statement. I thought it is for correct statement. That is why I gave the answer as A and B. Actually, that is not the correct answer. They have asked for incorrect statement. So 2 is your answer. C and D are wrong. Okay, so 2 is the answer. I made a mistake. They have asked for incorrect statements. Okay, so C and D only. Then next one. The following statements describe a few basic features of the interferometric reflectance imaging sensor used as a biosensing platform. So interferometric means it involves interference of light waves. Okay, that is what is meant by interferometric. It involves interference pattern of the light. Now this is a biosensing platform, and as I read each of the options, I will explain to you then and there, because that itself is giving the principle behind the interferometric reflectance imaging sensor. Okay. Now coming to the first statement. The biosensing platform is capable of high throughput multiplexing of protein-protein, protein-DNA, DNA-DNA interaction. Now what does that mean? So interferometric reflectance imaging sensor, it is a label free biosensing platform. Label free means it doesn't use any fluorescent probes or radioactive labels for detection. Okay, so it is label free platform. And what does this high throughput multiplexing mean? That means this IRIS biosensing platform, that is what is the short form of interference reflectance interferometric reflectance imaging sensor. That is the short form of iris, that is IRIS, all in capital letters. So that it can so this iris can analyze multiple interactions simultaneously. That is the meaning of this multiplexing of protein DNA and DNA interaction. Multiplexing means it can analyze multiple interaction. Which all interactions? Protein DNA and DNA interaction. And this because of this multiplexing capacity of this iris, it can rapidly screen various biomolecules. Okay. Now the next question is how you prepare this sensing surface. Okay, for analysis. So the sensing surface is prepared by robotic spotting of biological probes that are immobilized on functionalized SiO2 substrate. So this is the substrate for your iris. Which one? SiO2 Si. This is the substrate. Now you have to make this substrate ready to act as a sensing surface. That means it should sense the light that you provide it. Okay. Only then you will get the interference pattern. Okay. Now and also it should also be able to analyze the protein DNA interactions. So to make the surface of SiO2 Si platform ready, you have to do some basic requirements for making it a sensing surface. So SiO2 substrate, it is chemically modified. Okay, so that is the meaning of functionalized. You chemically modify it so that you can allow for the attachment of biological probes. Now what are these probes? It can be your antibodies or even your DNA. So if it is antibody, proteins will bind. If it is DNA, either your protein or your DNA can bind. Okay. Now you make this surface ready for sensing. So you attach biological probes there onto this surface. You have not applied the sample till now onto the surface. Okay, it is only the probe preparation. You have probe preparation means you can attach your DNA or your antibodies. Okay, to make this ready to accept your sample. So here for the application of this biological probes, you use a robotic system. So this robotic system, it will deposit small and defined spots of the biological probes onto this functionalized SiO2 substrate. Okay, that's it. That is what the statement B means. So that's also correct. A is correct, B is correct. Now as biomass accumulates on the substrate surface, a change in the interferometric signature occurs and the change can be correlated to a quantifiable mass. Now what is happening here is light is directed onto this SiO2 Si. So first you need a control where you have not applied your sample. So onto that light is directed onto this SiO2 Si substrate, and a portion of the light will be reflected. That is seen here. A portion of it will be reflected and it will result in an interference pattern. Now after that you record this data. So you get one interference pattern. Let it be the blue curve. Okay, the blue curve seen in the graph. You get that. Now you apply your sample. You have already made the surface ready by spotting biological probes onto it. Now you are adding your sample on top of that. Now this sample, it can go and bind to the biological probe according to their affinity and all. And this binding, okay, this binding of your sample will alter the thickness of this SiO2 layer. See here, biomass application, they have shown it a bit thicker than this one. Okay, so the biomass is nothing but your sample. Okay, the sample contains various molecules like your proteins, your DNA, etc. So it is it is altogether, it is called biomass. So application of this biomass onto this layer will alter the thickness of this layer, and it will cause a shift in the interference pattern. So that is the red line you can see in the graph. Okay, that is the shift. So here the reflectance pattern have changed. See here, the reflectance is like this. Here it is biomass application, thickness has increased, reflectance has changed. So when you plot the graph of reflectance against wavelength, you will get a phase shift. So from this phase shift, you can get information about the presence as well as the amount of molecules that are bound to the probe. So all these you can get from this phase shift data. Okay, now read this statement. As biomass accumulates on the substrate surface. So here it is. The biomass is keeping on accumulating, and this is your substrate surface. A change in the interferometric signature. So this is the phase shift in the interference pattern of the light and the occur, and the change can be correlated to a quantifiable mass. That's what I told you. And this change in the phase shift, it can give you an idea about the amount of molecules, or I can say quantity of molecules that are bound to the surface. That is what statement C tells. Now next statement, using this technique, picometer changes in biomass can be detected. Now iris biosensing device, it will not detect picometer changes. It can detect only nanometer changes. Picometer is still smaller than the nanometer, but it cannot detect that. So D is wrong. So which are the correct statements? A is correct, B is correct, C is correct, D is not correct. Then next one. Two newly identified proteins X and Y are tested for sequence-specific DNA binding activity. The results of an electrophoretic mobility shift assay EMSA with a labeled DNA fragment and proteins X and Y in various combinations is shown below. Okay, fine. Now see the principle of EMSA here. So you have the DNA, which is your target DNA, and it is radio labeled, and then you have added protein which will bind specifically to that target DNA, and you conduct the experiment, and you got a gel like this. Now what do you infer from the gel? So this is these bands of DNA, they are your labeled target DNA which are not bound to the protein. But once you add the protein, they start binding. So there is a difference in the binding pattern upon binding of the protein. So that means if the protein is bound, the DNA will run slower, and if the protein is not bound to the DNA, it will run faster. So when you see the gel, you can see a shift over there in the binding pattern. Now even if the protein was bound to DNA here, it didn't give a band here. I will tell you the reason slowly. Please wait. Now there is something called as super shift. Now for easy understanding, I am telling you, you add a DNA, you add protein, you got a band here. This is the normal shift. Now to confirm, you again add antibody to the which will bind to the protein. So again more protein molecules bound to the DNA. it will move slowly. So there will be what is called as super shift. Okay. Now it need not be always an antibody that you are going to bind to this DNA. It can be another protein which have the affinity for the same binding region as the first protein. It can be your second protein. So you add first protein, you got a shift here. Then you add a second protein, you will get a super shift. Okay, so it can be that way also. Now see the question. They have told something called as poly-DL-DC. It is a DNA duplex of inosine and polycytosine. That is what they have mentioned. Now I will explain to you what is this poly-DL-DC means. It is a non-specific competitor. Now when you conduct EMSA, you have got two competitors. One is the non-specific competitor and the other is the specific competitor. Okay. Now poly-DL-DC or a salmon sperm DNA, they both act as non-specific competitor. Now what is this non-specific competitor? So it is like a bogus DNA. It doesn't have any nucleotide bases which are similar or same as your target DNA. Okay, it is entirely different DNA, a bogus DNA. So what you do is, this is an unlabeled DNA. This poly-DL-DC, it is an unlabeled DNA, whereas your target DNA, it is labeled. So it is having some unrelated sequences and you add it to your sample. So in the sample, if there are some proteins which bind to any DNA molecule, they will bind to this poly-DL-DC. If you didn't understand, I will give you an example. Suppose you have a crude protein sample. It is not purified, that is why it is crude. Now this crude protein sample, it can contain many different proteins. There may be some proteins that are specific for your labeled DNA. To your target DNA. They will bind specifically to the target DNA. Now there may be some proteins that has got a tendency to bind to any DNA sequence. They just want to go and bind to any DNA sequence. So to minimize that, okay, to minimize this non-specific proteins binding to your labeled DNA, you add this non-specific competitor. So these proteins which has got a tendency to bind to any DNA sequence will bind to this non-specific competitor DNA. So their concentration in the crude protein sample will be minimized and the chances of your specific protein binding to the target DNA will be more. Okay, that is why you add non-specific competitor. Now there is an order for addition of the non-specific competitor in an EMSA. So first how you add is that first into the reaction mixture containing this crude protein sample, you will add first itself the poly-DL-DC non-specific competitor. So if there are any non-specific proteins which are found, they will bind to this DNA and they will not bind to your target DNA. Okay. So first itself you will add that. After addition of this non-specific competitor, you will add your labeled target DNA. Okay. Now coming to the specific competitor and why you didn't get a band over here. So specific competitor, they are also unlabeled DNA, but they have the same sequence as your target DNA. Okay, so specific competitor are also unlabeled, but your target DNA is labeled. And these specific competitors, they are used to confirm sequence specific protein binding. Okay, sequence specific protein binding by competing with the labeled DNA. So what you do here is, this specific competitor, it will compete with the labeled DNA for binding of the intended protein, and once the intended protein gets bound to the specific competitor DNA, it will result in a decrease of band intensity or disappearance of the shifted band if the interaction is specific. That means first in this gel, you in this lane you added your protein, your band got shifted. Now you want to confirm that it is because of the specific the intended protein binding to the specific region of the targeted DNA. You want to confirm that. So for that, what you add? You add a specific competitor. It is non-labeled, but it has the same sequence as that of the target DNA. You add that these proteins, which are the intended proteins, they will compete with the specific competitor DNA will compete with the labeled DNA, target labeled DNA for binding of this particular intended protein, and all the proteins that are present in the sample will bind to this specific competitor, and this will result in the gel showing no band in that region, confirming that the shift here is due to the binding of the intended protein to your DNA sample. Okay, so that is why you use specific competitor. Hope you understood. Then here also this unlabeled specific competitor, it has got a specific order of addition. So first I told you, you add the non-specific competitor, that is poly-DLDC, to the crude sample. Then you add the specific competitor, and after that you add the labeled target DNA. Okay, so that is the step. Now come to the question. So if you see the first line, non-specific competitor is not added, X and Y not added, only DNA is added. So this is your free DNA. It is not bound to any proteins. Free labeled target DNA. Then you add X, but you don't add non-specific competitor, you got a band here. Then you add Y, you don't add a non-specific competitor, you got a band here. Now you add both X and Y to DNA without addition of non-specific competitor, you get two bands as well as a super shift. Now why did you get this super shift? X and Y got bound to this DNA again. So X and Y can bind to the same target DNA. Okay. That's why you got a shift here, because protein concentration got increased. I mean the number of protein molecules bound to DNA increased. So so you got a shift there. That is super shift. Okay, this gel is showing super shift. Then you add poly-DLDC and you add X, but you didn't get the band for X. Why? Because I told you poly-DLDC, it is a non-competitive inhibitor, and it is an unlabeled nucleic acid, and it has some non-specific DNA sequences, and it is added to minimize the binding of non-specific proteins to the labeled target DNA. So in this lane, X band is not seen, so it must be a protein that will bind to any DNA sequence, and it is a non-specific protein. So that is, it does not bind in a sequence specific manner. It can bind to any DNA sequence. That is why it got bound to the bogus DNA. So now you know why you add non-specific competitor. Okay, now in the next lane you add Y, but without X, and poly-DLDC also is added. But still then Y was faithful and it got bound only to the target DNA. Labeled target DNA only it has bound. It didn't bind to your bogus DNA. Poly-DL DC, that is your non-specific competitor, it didn't bind. So Y binds in a sequence specific manner. Okay. Now read the options. Poly-DL DC is a DNA duplex. Which one of the following option represent the correct interpretation of the result obtained? Both X and Y are sequence specific DNA binding proteins. I already told you based on these two lanes, X is not sequence specific, but Y is sequence specific. That is why it got bound to the labeled target DNA even in the presence of this bogus DNA. So 1 is not correct. Then next one, X does not bind to DNA and Y bind specific DNA sequence. X binds to DNA. See here, you got a band over here when you add X to DNA. If you add X and Y, you got a band of X here. So that means X does bind to DNA. So 2 is also not correct. Both proteins bind to DNA but Y binds in a sequence specific manner. That is correct. I already explained that. X competes with Y to bind the same sequence. That is also not correct. X doesn't compete with Y to bind the sequence, but here you see you got a super shift band showing that X and Y both are bound to DNA. So there is no competition between X and Y. So 4th is also not correct. Then next one. A student used 420-mer oligos to amplify DNA. Okay. Using a regular Taq DNA polymerase from a wild type, a homozygous mutant having a deletion of the gene del. So this is the gene. So homozygous mutant will have this region deleted. That is, this 50 kb region will be deleted. And from a heterozygous mutant. Heterozygous means one allele will have the wild type and the other allele will have the deleted one. So as shown in the figure. Agarose gel electrophoresis profile using all 4 primers simultaneously on each template are shown below. Which one of the options given below represent the correct profile? Okay. Now see here. So this is the primer FW. It can amplify with the primer R and give a product. Primer FD can act as forward primer and D can act as reverse primer and it can give this product. Primer FW and this FD, they can act. Okay, they cannot. They are both forward primers. Okay, so it can be various combination of this forward and reverse primers that you get. But once you see the option, you see that the largest band size is 1000 base pair only. But here you can see 15 kb gene. That is not seen anywhere in any of the options. So this primer combination, that is primer FD and primer R, they have not taken. So how did you get this 1000 base pair in all the options? So 1000 base pair comes from this. Primer FW and primer D. See here, this is 500 base pair, this is 500 base pair. They both add up together and give you 1000 base pair. So here, if you see here, it is 1000 base pair. So FW plus D, you get 500 plus 500, 1000 base pair. Now there is a 750 base pair band, or it can be even more than 750 base pair. So from where did you get that? So that could be because of a combination between this FD and D, these two, this region. So that is a 500 base pair band. So 500 base pair. So this band over here you are not going to get with the wild type. Okay. So 1000 and 500 base pair are the ones that you get with the wild type allele. Now you see for the heterozygous, I told you heterozygous will not have this 15 kb deletion gene. It will be deleted in the heterozygous condition and one allele will have the 15 kb deletion gene in it. So both these bands you have to do PCR analysis. So here when you do PCR analysis, you find that for the wild type allele you will get 1500 base pairs. Natural. See here you got a heterozygous, you got 1000 base pair, 500 base pair, 1000 base pair, 500 base pair. Here you got 4 bands, again here 4 bands. Okay, we will see. Now once you delete this region, this whole thing is gone. So these two primers are of no use. FD and D are not of no use at all. So when you delete this region, this comes near to this 500 base pair. So you get something like this. So see here 500 base pair and then a 300 base pair. So here is the reverse primer R and the forward primer FW. They both will amplify and you will get a 500 plus 300, that is 800 base pair. So that is the band over here. Here you see the same band in heterozygous. Here I don't know how they got 4 bands. So that makes these two options wrong. So this is also wrong option. And this is also a wrong option. We have to see between these two. Then again they have told it is the homozygous one. So homozygous means this 15 kb deletion gene will not be there in both the alleles. So what do you get? You get only a 800 base pair band. 500, 300. This region not there. So both will give only a single band, that is 800 base pair. So here if you see you got 2 bands, which is not correct, but here only one 800 base pair band. So that is the correct answer. So answer here is option 2. This is also not correct. So that's it. Thank you.